Similarities are really in right now in the technologies. So you still need the media, you still need bioreactors, you still need to process cells. Technologically, it's very similar. The main differences are the economic aspect to it. And clearly, biopharmaceuticals are produced for essentially well-developed economies. Unfortunately, in many ways, they are only really available to the West. And of course, cost is not the real driver with a biopharmaceutical. Everyone would like cheaper drugs, but with our current range of biopharmaceuticals, cost has not been the big driver in their development. Clinical efficacy is clearly the driver, and quality. The challenge to me is to apply those very high-tech methods and techniques in an industry where clearly the product needs to be sold at a much lower price and also needs to be sold in much greater amounts and to a much greater range of people.
Welcome back to the Smart Biotech Scientist. I'm your host, David Brühlmann, and today we're exploring uncharted territory with Richard Alldread, who is the Chief Technology Officer at the Cultured Hub, a joint venture between Givaudan, Migros, and Bühler in Switzerland. After leading process development at CSL Behring, Lonza, and the National Biologics Manufacturing Center, Richard is now tackling the ultimate scale-up challenge: feeding the planet through cultured food. Let's dive into this fascinating transition. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Richard, to the Smart Biotech Scientist. It's good to have you on today.
Thank you. Honored to be here.
My pleasure. Richard, share something that you believe about bioprocess development that most people disagree with.
Okay, well, this was the hardest question to answer, really, because if it's something that most people disagree with, then I don't have such a big ego as to not think that maybe it's me that's wrong. But one principle I would very much adhere to is that bioprocess should be simple. As with many things in life, there's a great temptation these days to throw lots of very interesting technology, expensive technology at things, almost limitless process analytics and so on. But I think a bioprocess should be more akin to growing a plant than keeping a patient on life support. And for me, an ideal bioprocess would be one that does not require any process analytical technology. It's inherently robust that you know it will run and work.
I like that outlook, a process that doesn't need any PAT. Wouldn't that be amazing?
Yeah, I think you could argue potentially some CHO cell processes are getting close to that, but we're all scientists and we all like technology. We like to apply lots of interesting techniques to things to see what's going on, but which is great just as long as we're aware of what we're doing. And really, we should be aiming processes that don't need such close control.
Before diving further into the nitty-gritty of science and technology, Richard, let's talk about your story, because from pioneering life-saving biologics now to revolutionizing the future of food, that's quite a journey. So tell us, Richard, what sparked your first passion for biopharma, and now what ultimately led you into, shall I say, the dark side of things, into the cultivated food space, and what were some interesting pit stops along the way?
So, I mean, really, I guess for me, it all began many, many years ago when my parents used to subscribe to a magazine that had various articles in it on a wide range of subjects. And I remember being a voracious reader, reading an article about genetics and DNA. And that sparked a real interest. And so led me to studying biochemistry and molecular biology, without really understanding that there was such a thing as a biotech industry, even more a biopharm industry. But I went on and did my PhD, and I did much of my PhD in Manchester, UK, but also spent a lot of time working at a place called Porton Down in the UK, which was a government-run research establishment. And there really I was exposed, to some very inspiring people, but also some of the early days of biopharm and biotech and how we could use biotech, how we could use recombinant technology particularly. And then through subsequent job moves, became increasingly exposed to biopharm, to uses of biopharm, different types of culture, starting in bacterial protein expression and moving through insect cells and eventually into mammalian cells. So it was never really a big fan there. It was just a fairly natural way to develop, I think. A lot being interested in the biology behind it all, and also 20-something needing to start earning a reasonable living. It was a good way to go. And I guess I think I was probably lucky timing wise in many ways. I think I came into my degree and my PhD when recombinant technology had really become commonplace and a PhD student could carry out their own molecular biology. And then I moved into industry very much at the time of the big boom in monoclonal antibodies, which I think went on to really help the industry develop in a big way. I think a lot of what we think about when we think of bioprocessing now, particularly in animal cells, is really developed from a lot of that work in monoclonal antibodies. I'm lucky to work for some good companies, particularly giving a bit of a shout out for Switzerland, working for Lonza for many years. And of course, they were very much central to the whole monoclonal antibodies and biopharma development, as were their forerunners in the site I worked, which had been Celltech originally back in the UK. So it seemed a fairly natural evolution to me without never been a great one for having life plans. So it led to the biopharm industry. And then, of course, becoming exposed to all the types of therapies with various jobs, different antibody formats, different types of protein, gene therapy, cell therapy, and so on. And it's a fascinating world. The move to the food industry, again, was not particularly planned, other than it was an opportunity that presented itself out of the blue. It was a particularly interesting challenge to help design and build a facility for cultured food or cell cultured food production, thinking about what the challenges were for that, and to really apply those technologies, techniques, methods that had been developed in biopharma, but apply them to a different industry with different priorities. So in many ways, I don't sort of feel I've really changed what I do. There are just different emphasis taking over, but it's still very much technologically based on what I've kind of always done.
So, Richard, I'm curious because since you have seen both worlds and you just mentioned that what you're doing now is not necessarily very different. So what are the key similarities and what are some differences that between cultivated food and more established biologics?
So I think the similarities are really right now in the technologies. So you still need the media, you still need bioreactors, you still need to process cells. Technologically, it's very similar. The main differences are the economic aspect to it. And clearly, biopharmaceuticals are produced for essentially well-developed economies. Unfortunately, in many ways, they are only really available to the West. And of course, cost is not the real driver with a biopharmaceutical. Everyone would like cheaper drugs, but with our current range of biopharmaceuticals, cost has not been the big driver in their development. Clinical efficacy is clearly the driver, and quality. The challenge to me is to apply those very high-tech methods and techniques in an industry where clearly the product needs to be sold at a much lower price and also needs to be sold in much greater amounts and to a much greater range of people. So to me, technically, we have very similar challenges, but in the economics is where the biggest difference has come. I would say another big difference as well is, I think, the biopharm industry, and I've seen this very evidently over the last sort of 15 years, has moved towards quite well understood platform type processes. And they're largely absent in what we're doing now in the food industry. Again, a lot of what we think of as bioprocessing for biopharm purposes has been based around CHO cell processing, some other cells now for virus production and so on, but largely around CHO. In the food industry, we're dealing with a very wide range of cells of different types, primary cells, stem cells, immortalized cells from a wide range of different animals and not just mammal. I still call it mammalian cell culture and have to stop myself because a lot of the interest we see is not mammalian, it's avian and also fish as well. There's a big interest in culture of fish cells. Some very quite obvious differences when you think about it, but hadn't really occurred to me before, such as fish cells tend to grow in much cooler temperatures, which at least I suppose saves you on a little bit on energy costs of keeping the reactor at 37. So that's really the big difference to me, is lots of different cell types, lack of platforms and the need to produce at a much lower cost. I guess the other big part, of course, is that when we think of bioprocessing, easy just to think of the bioreactor, but of course, in a biopharm sense, a lot of the process is downstream, and we don't necessarily we have downstream processing in the food industry, but it's very different to the standard column chromatography filtration type approach of the biopharm industry. And of course, it's the biomass itself that is the product in the food industry, generally not a recombinant protein.
Speaking of economics, as this is one of the big difference between the two, can you give us some numbers for those who are not familiar between pharmaceutical production and the food? Like, what is the range of production cost and like, how does this compare? What is about the multiple of these?
That's a very difficult one to answer. And I think the feeling is that for food production, we will have to come down to no more than tens of dollars per kilo of product. Now, difficult to compare directly to the pharmaceutical industry or biopharmaceutical, as normally it's a cell product that is being produced. But typically recombinant proteins would be produced at thousands of dollars per gram. And so from a similar size bioreactor, the costs have to be so much less, orders of magnitude less for food production. And also the scale, I think, is a big influence here. Very difficult to give a definitive set of costs. We all know what food costs in the supermarket, and it's increasingly expensive, but it's nowhere near the sorts of prices we see for the latest biopharmaceuticals or even fairly long-standing biopharmaceuticals.
Yeah, the margins are definitely much lower in the food business compared to pharmaceutical, yeah.
It's not just the cost as well, it's the profit margins. You know, we all have a limit to what we will pay for food. And of course, I don't think that the situation hasn't been properly worked out for food production yet. People know that currently the processes, even the most advanced ones, are still really too expensive to hit the mass market. And there's a lot of work done to try and get those costs down. But where they will get to is still open to some debate, whether it can ever really. My view is it'll be difficult, certainly within our lifetimes, to compete with the cheap end of the food business. But what I think it can do is produce certain high quality ingredients for use in more value added products. And that's what we're seeing at the moment. Some of the products being targeted are the higher price ones. We are seeing people attempting, particularly seafood and the like, which is a premium food market compared to McDonald's chicken nuggets and so on.
So given the different process economics and also the lack of platform technologies in the food business, how does a process development / scale up look different? And also, what are the strategies you're using to bring down the cost significantly?
So, I mean, clearly scale is going to be one of the big strategies to bring down cost. And for instance, in the biopharm industry, maybe 20,000 liters was the biggest sort of mammalian process. But the emphasis in biopharm really has been to reduce the scale wherever possible, particularly with the move to single use items. The food industry is talking about needing potentially much bigger type facilities than that. Whether that's through scaling reactors as such or simply building multiple reactors is, again, open to discussion at the moment. There are certain problems that come with just building bigger and bigger reactors. It will give you even more scale-up issues. I think one of the developments that needs to happen is rethink how cell culture plants look. So there are certain givens for a cell culture plant. You need to maintain sterility, whether it's for food or for biopharm. Otherwise, you don't have a viable process. But right now, a lot of the food products are being developed in essentially biopharm standard equipment and plants. And that's something that will probably need to be questioned. The other big area is media. Then there's an awful lot of work at the moment on making media cheaper. There's been a lot of new companies get involved in the media business, not from the traditional biopharm suppliers, but companies involved with animal feeds and food ingredients and so on. And it's been good to sort of question, do we need media made to the same standards as we do for biopharm? And also to reformulate media. There's probably at the end of a CHO cell culture, there's an awful lot of nutrients in the media that we throw away. And there's been a lot of work done, or is a lot of work ongoing, to look at what are the exact nutritional requirements of cells being grown for food and how can we make best use of the media. So I think how we design and build plants, the scale we go to, and the media will have big influences on the price. I think process intensification, and I know that's probably shared with biopharm as well. There's a lot of interest in intensifying processes. That'll certainly be needed for food production. But again, combining that with the large scales will be a huge technical challenge.
Speaking about scale up, I'm just wondering, where are the limits? Because we have very different cell types. We have fish cells, we have chicken cells, we have beef cells, you name it. Some are cultured in suspension. Others are adherent cell cultures. So where do you see the limit? And when you say large scale, what is the scale actually we're talking about?
Well, I mean, at the moment, people are talking about going to over 20,000 litres in some instances. But again, I think it's such an immature market. Nobody really knows what are the potential scales. A lot of it will depend on customer demand for the products. So certainly in the last week, I've seen 25,000 litre reactors being proposed as a solution for some product. Back to your point about suspension culture versus adherent. I think in my view, it's certainly in the near term, we are talking about suspension cultures in order to keep the economics at a reasonable level. I think once you're into 2D cell culture, then it's very difficult you magnify those problems of scaling and scaling at a reasonable cost. And there's so much more steps involved and manual handling needed. So yes, sometimes it's a bit of a mixture with growth on scaffolds and microcarriers and so on. But I think over the next sort of 5-10 years, it's certainly going to be suspension culture in a fairly traditional way as we know it that predominates.
In the beginning, you said, let's keep bioprocessing simple. And in an ideal world, we will not use PAT. We all know that we are not in an ideal world, especially in the food business where it's very immature, very early. How do you think about digital technologies, about PAT systems or modeling or even AI, if we want to go even fancier? How does that play out in the food business?
I think they're all important. When I said the ideal process doesn't use PAT, I think we will always use PAT to develop the ideal process. My idea is that you don't need it routinely to run a final process. I think the more PAT we have available in order to understand what's going on in the process and to develop a more robust process is good. In terms of modeling, I think that's going to be hugely important for all the bioprocessing. And I still think we are not great at it compared to other industries. And some of that is understandable. We're dealing with complicated, complex biological systems, and it can be difficult to model. One of the big problems of applied modeling in the food industry is back to that. We have lots of different cell types, and therefore the sort of platform data and what you can take from one process and apply to another can be very poorly understood at the moment. So I'm a huge fan of modeling. I know there are some small companies around Zurich who, have very impressive systems, but still, to me, applying it to novel platforms is a difficult thing. If you've got years of historical show data producing monoclonal antibodies, it becomes much easier to start modeling the process. And as for AI, like everyone, I hear that AI is going to change our lives completely. And I'm sure in some ways it will, but I'm probably not the best person to give you a very educated answer as to what the impact of AI is. Clearly, it's going to be big across everything we do. But maybe getting too old, but I still think that good scientist, a good bioprocess engineer is always going to be needed to develop the process. And yes, he can make use of all the other tools, but I don't see the need for skilled scientists, technicians and engineers is going to go away soon.
And that wraps up part 1 with Richard Alldread, where we have explored the fundamental differences between biopharma and cultured food development, from economics to cell line challenges. In part 2, we'll dive into the Cultured Hub's unique approach and the massive scale-up innovations needed to feed the world. Do you have process development questions? Book your free consultation at BrühlmannConsulting/call, and let's solve these problems together. Thank you so much for tuning in today. Don't forget to leave a review on the platform you found us on, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.